Hi, everyone. We're students from North Tama High School, and we were selected to take part in an exciting project called Translating Information to Demystify Advanced Biomanufacturing. This project is funded by the Education and Workforce Development Seed Funding Program for Chemurgy 2.0 through Iowa NSF EPSCoR. The goal is to help make information about advanced biomanufacturing more accessible to the general public, especially the research being done right here in Iowa, and why it matters for our future. The project we choose to explore is called What Are Nanobodies? We're Tyrus, Nathan, and Johnathan, and we're excited to walk you through what we learned. This project was led by Dr. Lisa Chizek from Iowa State University and supported by Dave Chizek, our project volunteer.
So, what exactly is a nanobody? A nanobody is a really small protein that can bind or stick to other things with super high accuracy. Scientists say this is high specificity and affinity. And if you're wondering, affinity basically means how tightly it sticks. Bret Lange, a researcher at Iowa State, is working on a project to engineer nanobodies that bind to a protein called fibroblast activation protein, or FAP for short. FAP is important because it shows up in higher amounts during diseases like cancer. So if we can make nanobodies that stick to FAP, we can use them to detect or even treat diseases in non-invasive ways.
Bret's research uses something called cell-free expression, or CFE, to test nanobodies faster and more efficiently. Traditional protein production takes place in living cells and can take weeks, but with CFE, Bret can do it in just one day and test way more protein sequences at once. This faster method helps Bret figure out which nanobodies have high affinity for FAP and which ones don't, so he can filter out the weak ones quicker and move forward with the promising ones.
Using the CFE method, Bret can rapidly generate nanobodies and test which ones stick best to FAP. The diagram here shows the workflow of how nanobodies are screened using Bret's process. It's fast, efficient, and really changes how researchers approach this kind of work.
So how does CFE actually work? Bret mixes together three key things: an extract, which is kind of like the machinery harvested from cells like E. coli, a supplemental mix, which contains extra minerals needed for protein production, and DNA, which has the instructions for making the nanobody, including mutations to test different designs. Then he incubates the mixture overnight, and the next day he has new nanobody proteins ready to test. This is a photo of the incubator and shaker used to grow the cells needed to make the extract.
Once the nanobodies are made, Bret uses a tool called the Blitz to measure how well the nanobodies stick to FAP. This method is called biolayer interferometry, or BLI. It tells you how much of the nanobody attaches to the FAP and how much falls off. These two things, called association and dissociation, are used to calculate the overall affinity. The stronger the affinity, the better the nanobody sticks.
After testing, Bret shares his results with other researchers. They use his findings to design even better nanobodies and then send them back to Bret to test. It's a cycle: design, build, test, learn. Over time, this loop helps scientists create nanobodies with super high affinity to FAP. This design loop is key to improving diagnostics and treatment options in the future.
This research is important because proteins, like nanobodies, have many uses. In diagnostics, nanobodies can stick to FAP to show where something might be wrong in the body. In therapeutics, they can carry drugs right to the problem area, like a targeted missile. There's even potential for nanobodies to be used in other areas, like breaking down plastics, so the possibilities go far beyond just medicine.
Bret Lange is the researcher behind all this work. He studied biochemistry at the University of Nebraska and is now in his second year of a PhD in chemical engineering at Iowa State University. He's always been fascinated by DNA, how just 4 chemical bases can create all life on Earth. His big question is, can we design proteins to do whatever we want? That's what drives his research.
If this sounds interesting to you, here's what it takes to work in this kind of field. You should be curious, persistent, even when you fail, okay with not having all the answers right away, and willing to figure things out on your own. To prepare, take classes that make you think critically, try out research opportunities, And most of all, just dive in.
Finally, we want to give a big thank you to Bret Lange for taking the time to answer all our questions, show us around his lab, and explain his research in a way we could understand. We learned a lot from this project, and we hope we helped you understand the world of nanobodies and biomanufacturing a little better too. Thanks for listening.